Okay, good morning, everyone. So happy Friday to you. Before we start with today's lecture, I've invited Juliana, who is one of our TIs this semester. Juliana is also involved with the pre-health, pre-law advising committee that's available, PAC, which is a wonderful resource here on campus for helping fellow peers who are in life sciences sort of decide upon different decisions or pathways through their courses. And so I invited Juliana to spend a couple minutes at the start of this lecture to talk a little bit more about PAC. You know, earlier this week, I had mentioned that we have a molecular cell biology BS program. And I was very happy to see that some of you emailed me about that because you didn't know we had such a program. And now that we're like a third of the way into our Bio 275 semester, you're very interested in finding out more about that program. So I think it's very nice timing for Juliana to just say a few words about PAC and to give you information about if you want to go ahead and contact PAC and make use of their services. So Juliana, would you like to say a few words?
Yeah, so like Dr. Shen said, I'm a TI for 275, but I'm also a pre-health peer advisor for PAC. So what we do is we help students who want to go into a career in health or law and figure out, how to get into these professional schools and how to be successful in your pre-health pathway. So if you're interested in going into something like medicine, pharmacy, physician assistant, dentistry, can you use the reactions button in Zoom to do like a little thumbs up really quickly? Okay, I see a few. I'm sure there are more. Okay. So for everyone that put a thumbs up, our advisors can help you. So there's several things that we can help you with. I think the most common are like academic plan, experiences, entrance exam, and application, right? So figuring out, do you have all the prerequisites that you need to get into something like medical school or physical therapy school? Talking about what experiences you can involve yourself in and how to find those opportunities. So research, volunteering, shadowing. Do you know when to take your entrance exam? Do you have an entrance exam that you need to be taking? 275 is important for the MCAT. We actually recommend that you take it for the MCAT if you do need to take that. So it's important that you know when to take it, what is even being tested on it, and how to study for it. So our advisors can kind of walk you through that process. And then lastly is the application, right? So are you familiar with the application timeline? Because it actually, for most schools, it takes about a year to complete the application cycle. So, you can't just apply this semester and then get in the next semester. It takes a whole year. And so you have to prepare ahead of time. And that's what we're here for, is making sure that you apply at the right time to get into your schools at, when you want to get in. And then once you're ready to apply, our PAC director, Dr. Kiana Sharoma, she meets with everyone, or everyone that meets with her, to go over their application, their personal statements, and do mock interviews. So there's a lot of things that we can help you with, even more than what I talked about. We meet with students that don't even know what field to pursue, and that's okay. If none of that interests you, we also have a workshop series that just started this week, where we provide different workshops on how to apply to these professional schools. And we also have a medical student panel. And I think we also have like a mock MCAT. So if you're interested in those, you can email us. You can check out our website. If you just Google UH-PAC, I think we're like the first one to show up. So I'll also leave a link in the chat and the flyer for that information. On the website, you can also find information to schedule an appointment with an advisor. We are working remotely, and so all of our appointments are done through Zoom, but we would be glad to meet with any of you. But I will leave the information in the chat. But does anyone have any questions for me at this time? Okay. So I just left the information. We have our email. We do have an Instagram and then our website. Feel free to contact us if you have any questions. But I look forward to hopefully meeting with some of you in the future. But yeah, thank you, everyone. And then thank you, Dr. Shen.
Okay, thank you, Juliana. Thank you for that nice presentation. So as Juliana said, maybe one of the easiest ways, if you lose track of the info that she put in the chat, is just to go ahead and do a Google search, right, and find the UH-PAC website that way. I remember when I was an undergraduate, especially early on, You know, I really wish that there were peers and other people to help me sort of guide my way through my undergraduate studies, because a lot of it was just me sort of stumbling along, right, and just finding out through friends what I was supposed to be doing or how I was supposed to be preparing. So here, I think the PAC represents a wonderful resource of not only faculty but also peers to help you feel more comfortable and confident that the things you are doing are the things that are the right steps to take as you look towards graduating. All right, good. Let's go ahead and start in with our lecture. So for the next 3 lectures, we will be talking more about this concept of gene regulation, right? Remember, gene regulation is how different cells of your body know which sets of genes are the ones that are relevant for them to be expressing and using. Right? And we will look at different levels of control that your cells have over influencing what genes are ultimately made into proteins in order to define that particular cell and determine what that particular cell's job is and what it is capable of doing in the tissues of your body. Okay. So this looks like a very busy overview chart, but it is just an overview chart. Okay, so I don't want you to be freaked out about this. What we will be talking about in the next few days are these different levels of how genes can be controlled to determine ultimately whether things like proteins are made for them. So there are 5 levels. One is just at the level of the genome itself. In other words, remember, the genome is our DNA that's packaged up in the form of chromatin, right? That makes our different chromosomes, right? The individual DNA molecules that are the chromosomes in the human genome. And we've already learned that the DNA itself can kind of be modified or handled in a certain way to control access to the genetic information contained within. Right? On the right part, you know, I've just put in some little illustrations here to help you remind you when we're talking about these different levels, what we're discussing, right? So at the level of the genome, you can control how the DNA is organized and whether different types of proteins like RNA polymerase have access to the DNA, ultimately determining, you know, by the end of sort of like this first level here, whether a gene is available for expression. Right? Then the next step would be transcription. When the RNA comes in, RNA Pol II comes in and starts making a transcript from, let's say, a gene, right? That would result in a primary RNA transcript being made. The third level would then be once you have that primary transcript, right? It's over here that has the entire, it's like a photocopy of the gene made in RNA form, right? You would then process this, maybe cut out the parts that you're not going to need. So cut out the introns, leave the exons to make your final mRNA, right? So RNA processing and editing is another sort of third level where the cell has opportunities to influence whether something is going to be made from this. By the end of that, right, the mRNA is then moved out of the nucleus into the cytoplasm. So here the mRNA is now in the cytosol, in the cytoplasm, where translation takes place. So the ribosomes that we've been learning about have access to the finished mRNA and can translate the codon sequence in the mRNA into a chain of amino acids. And then you'd have a polypeptide chain. And ultimately, that polypeptide chain is going to fold up into its proper 3-dimensional structure as a finished protein. But we also learned that that protein then can also be modified in some ways to change its shape, maybe to function as on and off switches, right, that control how that proteins work. So that's another sort of stage or level at which the cell can control, right, how the protein not only is made, but how the protein actually functions. Right? So that's what we're going to talk about in the next 3 lectures. So today we'll start off just talking about the sort of stage number 1 here, the genomic level. So I think it's important for us to remind ourselves that the genome, right, contains all the information, all of the genes that are needed to define all the different complex functions that your cells do, also determining what different types of cells you create and how those cells use certain sets of genes to do their job. right? But let's step back and actually think about how amazing that is, right? That every cell in your body contains a copy of the master blueprint, right? And all of those instructions for everything that your body needs to do, right, is contained in that master blueprint. Let's think about how amazingly wonderful this is. Right? Let's start off with sort of this little thought experiment here. Let's say you wanted to build a machine, right? So here we have this imaginary machine that the whole purpose is you want to be able to take raw materials that are just floating in the air because it's like an endless source of raw materials, things like water that's floating around, carbon dioxide. You want to be able to somehow chemically react them together to make energy, right? And then you want to be able to store that energy in something like a battery. You know, and for kicks, we want to make this solar powered, because why not, right? Because right now we have an endless source of solar energy. So let's imagine if you or I were to try to build such a machine, right? It might look like some crazy cobbled together contraption of these different parts. Some things that take in air. You have all kinds of hardware that would then somehow funnel or direct this. It'd all be underneath some sort of automated, fancy computer control. There'd be a chemical reactor where these reactions take place, the carbons and the oxygens and the waters, the hydrogens from the air. And importantly, whatever energy that you make, you have to store it in a battery, right? And this whole thing is just run by solar panels. Now, I don't know about you, right? But I would have no idea how to build something like this from scratch. And hopefully, you know, even if you are more sort of engineering inclined than I am, hopefully you would accept, yeah, this would be a pretty fancy, complicated kind of device, right? But what would this machine look like if it was not this kind of crazy cobbled together contraption that our hands have made, right, with circuit boards and various hardware? What if this was actually designed by nature? What would something like this look like? I suggest it would look something like this, right? A carrot. And you might laugh and you're like, oh, it's a carrot, right? That's not a complicated machine, right? Yeah, it is, right? In fact, it's exactly the type of machine that we were just kind of having a little daydream about, right? A carrot is an amazingly complex machine. Think about it. This machine has parts that can harness the energy of the sun to drive the machinery. That machinery inside the carrot fuses water and carbon dioxide that it gets from the air to make sugar, right? A molecule that represents energy that is stored, right? Energy that is captured by the sun and turned into chemical energy. Importantly, the carrot can store this extra energy, right, in the wonderful, crunchy, sweet root of the carrot. That's its battery, right? That's where it stores energy to use for some other time, right? And even more amazing, this carrot can do things that our clunky machine that we put together by hand cannot do. This carrot can grow, right? And this carrot can reproduce. Right? That's pretty fantastic if you think about it, right? All of these things are a function of the genes that are contained within the carrot genome that provide all the instructions, right? How to make such a wonderfully complicated machine that can carry out these wonderfully complicated processes. So let's look at this. The carrot genome is about 473 million base pairs of DNA, and it is spread out on 9 separate molecules of DNA, right? 9 separate carrot chromosomes. And together that contains, we estimate, around 32,000 genes, right? 32,000 separate stretches of the DNA, right? On a chromosome or on the chromosomes that actually code for things like proteins that then function as the little machines that are making all of these wonderful living processes possible. I've shown you a genomic map similar to this when we looked at E. coli bacteria. Remember, the way these types of circular maps work is they usually will have the inner circle will be the actual DNA, the genome itself. In the E. coli, there was just one continuous ring because the E. coli bacteria just has one chromosome, right? Whereas this, you can see, is the carrot genome. So it's divided into 9 separate DNA strands represented as you go around the circle here, right? And as I told you last time, you don't have to really understand, you know, what all this stuff is in here. But I wanted to introduce and get you familiar with looking at something like this, because this is how we now, you know, when we're studying the genomes of organisms and gene expression, we oftentimes map them out this way, because each layer of data or layer of information as you go outwards represents some other type of event or phenomenon that's taking place that can be traced back to a particular location on one of the 9 caret chromosomes, right? So these little peaks and valleys might be different data readouts of things of, oh, are we getting levels of mRNA that are actually coming off of this region of this particular chromosome? In other words, the evidence that there's gene transcription going on certain spots, right? And then as you work your way out, you finally get to things like, here's where they're going to annotate or actually identify where certain genes actually are, right? So these red peaks here might actually represent, right, levels of a particular protein that are expressed on, in this case, chromosome 5, right? So you're able to, because of the way this map is laid out, trace it back, and you'd say, ah, right, this particular gene that codes for this protein that this caret cell was in the process of making protein from, right, is located here. Okay. So the caret genome, you know, that's what you're looking at here, about 32,000 genes on 9 chromosomes, right? But that's a caret, right? I often say, hey, you know, we give a lot of other organisms, we don't give them their due credit, right? We always think a fly is a fly, a carrot's a carrot. I mean, maybe some of you are going to eat a carrot for lunch today. I have carrot sticks in my lunch today to help balance with my potato chips. But anyhow, right? But what about something like more complicated, like a mammal, right? All right, since we're talking about carrots, let's talk about Bugs Bunny. So a rabbit's genome is made of 2.6 billion base pairs. So that's getting close to the human genome size, right? The human genome is 3.2 billion, right? So far more DNA than a lowly carrot, right? Bugs Bunny, the bunny genome is spread out on 22 chromosomes, right? So very similar or a lot closer to what we have. You know, we have 22 autosome chromosomes plus our pair of sex chromosomes, right? And a bunny's genome has about 25,000 genes. So in other words, it's right on par with the human genome in terms of the number of genes that have been identified. But look at this, right? The bunny genome and the human genome, we actually have fewer genes than the lowly carrot does, right? So again, this really forces you to kind of reevaluate how the relationship between number of genes, the size of a genome of an organism, and what we perceive as being an advanced or much simpler life form. Okay, so let's recall that every single cell in our body, just like every single cell in that carrot or every single cell in that rabbit, right, contains a complete master blueprint, a complete copy of that organism's genome. Right? So here we have our human genome laid out in front of you as a karyotype. So we have our 22 pairs of autosomes and our pair of sex chromosomes, right? In this case, this comes from a male. So the male inherited an X from mom and a Y from dad, right? And each one of these pairs represents one chromosome 1 that came from mom and one chromosome 1 that came from dad, right? I mention this not because I think this is new to you, because we certainly talked about this enough, but oftentimes it's confusing when you say what constitutes the human genome, right? If you look it up, depending on where you look it up, it will say something like, oh, 22 chromosomes and then a pair of sex chromosomes, right? Or you look it up somewhere else and it says, oh no, the human genome is 46 chromosomes, right? It just has to do with how you want to interpret it, because it all means the same thing, because each one of these, right, in a pair, like chromosome 1, just because you got it from mom versus from dad, it still is chromosome 1, right? So that's why it can get a little bit confusing how you describe it. And think about it, right? So if we say that A chromosome, you know, I've used this example before, is like a long cash register receipt, right, that has information on it. In this case, it's a set of different coupons on this receipt, right? So it's information that is usable. Remember, so in our genome, we wouldn't just have one of these receipts, one molecule of DNA, right? We would have 23 pairs of these, right? Each containing different sets of coupons. And these coupons may or may not be particularly of interest to you, right? So for example, you know, on this big longs thing, I see that there's a coupon for, here's $2 off. What is this? Gold emblem, nuts, trail mix, or salty snacks, right? I kind of like that, right? I'm a big fan of salty snacks. I think it's kind of funny. It's sort of generic. Like, yeah, I want $2 off some salty snacks. You know, further down on it, there's $3 off. What is this? Maybelline Cosmetics or Garnier facial skincare. All right. You know, so maybe I don't care about that, right? Maybe my wife really cares about that. I'm after the salty snacks, right? But they're contained on the same set of genes, right? They're physically on the same DNA molecule, but it might just be that I care more about using this coupon for the salty snacks, but maybe my wife cares more about getting $3 off Maybelline Cosmetics, right? So that's what we're talking about here. Even though a cell in your body has all of these coupons at its disposal, right, because it has the full set of your genome in it, right, all 23 pairs of chromosomes, a particular cell only needs to care about or is interested in using some of this information and maybe not other information. And that's what determines the different cell types in your body. And so again, you can either think of it as this represents the genome because this is chromosome 1, or you might, depending on how you want to define it, say, well, no, you have 2 chromosome 1s, right? So you might say, no, I prefer to think of the genome as being this, one that came from mom and one that came from dad, right? It's really just semantics, but it can get confusing if you don't make that kind of distinction. Right? This concept highlighted in blue here, genomic equivalence, is what we are talking about. All cells have the same genetic potential. All cells in your body have the same human genome in them, right? And that is kind of our starting point for thinking about how cells regulate the choice of which genes to use. So to demonstrate that, we can use 2 examples. Cloning, and then also our discussion of stem cells. So let's go back to our lowly carrot here. Right? We already said we have a newfound appreciation for carrots. Right? Now you feel bad about eating a carrot. Right? A carrot, like many plants, you know, one of the interesting distinguishing features that separate plants from animals is that the cells of plants have remarkable ability to regenerate the entire rest of a plant that's missing. So you can take, for example, a carrot. You could do this, you know, just with a blender or something, cut a piece of the carrot off, maybe blend it up into small fragments, and then put it in a tube with some very rich media that helps keep the cells happy, right? There's all kinds of things like amino acids, carbohydrates in there, right? So the cells are happy, and you can get an individual cells to kind of break apart and just be single cells floating around, right? So you can isolate these single cells that came from, you know, a random part of the carrot here. And if you put them in the right conditions where they have all the nutrients they need, right, they will start to divide. They will divide and actually start becoming an embryonic plant. At that point, you can take this, plug it into maybe some agar, right, that again has nutrients in it. So now this little embryo, it's almost like you've planted it, right, in some nutrient-rich soil in a way. The cells will continue to change their identity, and eventually you can grow an adult carrot plant from it again. And importantly, what? The genome of this adult plant is the exact genome that came from the original carrot that you chopped up, right, and got the cells from. So that means this adult carrot plant, even though it's a separate individual, is a clone. a perfect genetic copy of the original starting individual. So plants do this. Many types of plants propagate or reproduce just by cloning. I mean, you know this, you maybe don't think about it, but in your front yard, right? All the crabgrass or whatever, you know, the kind of grass where you see some tufts of grass, but then there's the long kind of stem that runs along the ground, the thing that sometimes you trip over if you're running, right? And then what? Different places along there are new little bunches of grass growing where roots have been set down, right? So plants are propagating by cloning, because then if those different strings get cut, you have individual tufts of grass, individual grass organisms, right, that are growing, but they're all clones of themselves, right? Because they have the same genetic, they have the exact same genome. Okay, and this works because the master blueprint, right, is always included, no matter what kind of cell in the plant that you're talking about. Just like in our bodies, no matter what kind of cell you're talking about, if it has a nucleus, right, it has your copy of the master blueprint in it. So with us, though, if you're talking about mammals, yeah, you can't do that. I cannot take, for example, like some skin cells from me, plop it into a test tube and do something like this, right? You know, or I don't know, can we? Right? Because conceptually, what's the difference? So back in the early or mid 90s, 1990s, was the first time that scientists successfully cloned a mammal, right? And this was in 1996, and that mammal was a sheep, right? And so this was the experiment that they did. So this was a group of European researchers. They took a cell, so a cell from the mammary tissue of an adult white sheep here, this fin dorset ewe, right? And they were able to tease the cells apart, and just like the carrot experiment, put them in some sort of culture medium, right? But in this case, what they did was they put it in medium that was depleted of vital nutrients, so that basically all these cells, they would stay alive, but they would stop doing their normal like mammary gland functions, right? So that means all the expression of different genes and things like that that would normally define these as a mammary cell kind of were erased, right, or blanked out, because under these conditions, the cells no longer wanted to actually carry out their normal sort of gene programming. Right? At the same time, they took an unfertilized egg cell from a different type of sheep, right? So you can see they on purpose chose the donor egg sheep to be very different than the cell that they got the mammary cell from, right? They did it on purpose because the whole point was they're trying to clone this white sheep on the left here. So they took that unfertilized egg, and you think about it, what is an unfertilized egg, right? It is a mammalian cell that contains all the proper sort of proteins and functions to actually start, you know, turning into a new individual. But what's the stumbling block? The stumbling block is there's only a haploid genome in there, right? So in other words, the egg cell only has one copy of the genome, whereas a full-fledged, right, individual needs to have two copies, right? One from mom and one from dad. Right? So until that happens, until the egg is fertilized by the sperm, right, to get the two copies, the egg is not going to start dividing. So what did they do? At this point, right, this unfertilized egg, they sucked out the nucleus. So all you're left with then is all that good stuff, right, proteins, nutrients, whatever, that an egg needs to start dividing, but you've taken the nucleus out, right? So what they did next was they took one of those mammary cells, right, that they were preparing over here that had gotten sort of stopped, so it stopped doing mammary cell functions, and they fused the two cells together so that now the full diploid genome that came from the white sheep on the left, right, now was the nucleus of the egg cell, right? And now you have a diploid genome inside of this egg. They then sort of zap it with some different sort of stimuli to get it to start dividing. and that egg proceeds to divide as though it's a fertilized egg, right? So eventually, once the embryo got large enough, they implant it into the uterus of a surrogate sheep to carry the developing baby lamb. And then Dolly the lamb was born in 1996, right? And once Dolly was born, they did genetic testing, and Dolly was a clone, the exact genetic makeup as the original sheep that they got the DNA from, right? And so this is all the way back in 1996. To this day, they've now gone on to clone all kinds of animals, including primates, right? So now we're getting into kind of scary ethical territory, because now they've successfully cloned things like chimpanzees, right? And evolutionarily, chimpanzees are about as close as you can get to people, right? So it's really fascinating because it raises all kinds of ethical dilemmas. And what was once science fiction, we're talking about, just 20, 25 years or so ago, would have been completely science fiction. Things you only see in crazy sci-fi movies is now right at the cusp of becoming reality. If we have time at the end of the lecture, I'll tell you a story about meeting the scientists who cloned Dolly. So let's think about this, right? Our body, you sitting there right now listening to this lecture, right? You have cells in you that still have the ability to turn into different types of cells. After all, most of the cells that you see are what we call fully differentiated cells. In other words, cells that have already carried out their genetic programming so that they've become exactly the type of cell they're supposed to be. Nerve cell, muscle cell, gland cell, etc., right? A red blood cell, something like that, right? That means they've made all these sort of decisions about what genes from the master blueprint they're going to use and what genes they're not going to use. Potentially ever again, they won't use certain genes, right? So the cells in our body that are constantly replenishing these fully differentiated cells are cells that we call stem cells, right? These are cells that, yes, they've made certain decisions to end up being something like a hematopoietic stem cell that's in your bone marrow, right? So this hematopoietic stem cell still has the ability to make further decisions about what sets of genes to turn on and turn off to enable it to eventually become something like all the different types of cells in your blood. White blood cells, cells that make platelets, or red blood cells, or immune cells, right? Things like T cells, B cells. Right? All of these important cells on the bottom that are part of your blood and an immune system, they all come from the hematopoietic stem cells that are hanging out in your bone marrow right now. Right? And we call this hematopoietic stem cell multipotent. Why? Because it can still turn into a handful of different fully differentiated cells, but this hematopoietic stem cell that's multipotent in your bone marrow cannot turn into something like a muscle cell or a nerve cell or a gland cell. Why? Because those decisions, right? The hematopoietic stem cell is already far enough down the line of making decisions such that it can no longer go another way and become one of those other types of cells. It can only make an additional set of decisions to become the blood cells that you see on the screen here, right? These fully differentiated or committed cell types. So embryonic stem cells, you've potentially heard about ES cells and ES cell research. So what are ES cells? Embryonic cells are human cells that come from human embryos. So when you have the fertilized egg that begins to divide and form, the zygote, right? So from one fertilized egg, you get 2 cells, then 4, then 8, 16, 32, et cetera, et cetera. At a certain point early on, these cells are still what we call pluripotent. Because think about it, right? Unlike the adult multipotent stem cell that hangs out in your bone marrow and can make more blood cells, right? But those are the only types of cells it can make. Way back in time, right, 20 plus years ago, you were just a ball of cells, like literally 32 cells. And those 32 cells eventually made or makes the 30-something trillion cells in your body. So that should lead you to realize, yeah, at this point in the decision-making process of what genes are actually being used in these cells, it's not really decisions that are already sending them down the path to becoming something like a bone marrow cell or something else. At this point, it's all about them just dividing, right? And ultimately, these early cells have the potential to become any cell in your body eventually, right? Again, because this is high enough up on sort of the decision-making process of gene regulation. Right? If you take some of these cells from the blastocyst here, like this blue one here, right? These cells are capable of infinitely dividing, and they have the potential to become any cell in the human body. That's why, right, modern medicine, there's so much interest in the applications of using stem cells to do things like replenish damaged or diseased tissues in people's bodies, tissues that normally can't heal by themselves. Things like spinal cord injuries, right? Because nerve cells do a very poor job of replenishing if they are damaged, right? But yet, the nerve cells in the spine of a paraplegic or quadriplegic individual, those originally came, right, from stem cells. So there's great interest in being able to use stem cells and maybe get them, to start making gene programming decisions to eventually turn into things like mature nerves. So of course, though, because these come from fertilized embryos, there is great moral and ethical dilemma in this, right? And that's why I'm sure you're aware that there's a lot of outcry and a lot of pushback from general public and the scientific community about embryonic cell research. Another approach, though, is if you think that the embryonic stem cell, right, and a fully differentiated cell in the body still contain the exact same genome, what's the difference? The difference is, right, in the embryonic stem cell, the genome is a blank slate, right? No decisions have been made yet on what genes are turning on and turning off. Everything is available. Whereas the fully differentiated cell, like a skin cell, right, from your arm, is already so far down a whole series of different decisions about what genes to turn on, turn off, that it is stuck just being a skin cell, right? But yet, all of the genes are still there. So one fascinating approach is, what if you could take a fully differentiated stem cell and reverse the process, reverse the decisions that were made along the way that turned it into a skin cell, and get it back to becoming more stem cell-like. Okay? And so that's what researchers are working on now, trying to make pluripotent stem cells in the lab. So what do I mean by that? Let's go back to our little exercise here, right? What we're saying is, in the genome, there are certain genes that might have been used very early on in the stem cells phase, right? And those genes controlled things like the stem cell being a stem cell, sort of dividing and proliferating, and that being its main job. Right? Let's say, for example, there's a coupon here, right? Yeah, let's just use, here's the salty snack coupon, right? The salty snack coupon, maybe in the stem cell, was widely available for the stem cell to use, right? Because maybe that's an important early type of developmental gene, or early type of gene that controls something like cell division. Right? As that stem cell divided and became more differentiated, maybe that coupon for the salty snacks, right, gets hidden. It's still present on the chromosome, but there are changes that have happened in the chromatin that now hide that coupon, right? So now the skin cell that's in your arm no longer has access to that early developmental gene anymore, right? It's been shut down. Right? So the secret is, how do you go ahead and wake up that gene again? Right? The truth is, you don't have to wake up that gene. What you can do is say, well, you have a skin cell where that gene is hidden, right? And we want that cell to start making the protein product of this gene again. How can we do it? Well, guess what? I have here a copy of the salty snack coupon, right? What if we just artificially put a piece of DNA into the cell that has that gene that we want it to start expressing again, right? That's what scientists are doing. Okay, so you can see here you have a fully differentiated cell, this purple one, maybe it's a skin cell, for example. And what you do is you put in pieces of DNA that specifically are the salty snack genes, no, right? That are specifically transcription factors that are the ones that early stem cells need to be making in order for them to be more stem cell-like. So here's 4 examples of these transcription factors here: OCT4, SOX2, KLF4, and CMYC. Right? Researchers have found that if you put this combination back into a fully differentiated cell, what these transcription factors do is they bind to the genes, right? And they start erasing different types of modifications that were made on those genes, so that the hidden genes that are all part of being an early stem cell can now be accessed again. Right? And in the process of doing that, the purple skin cell loses its skin cell properties and becomes more like a pluripotent stem cell, right? And then if you go ahead and now start that cell down the pathway of making different decisions, right? Instead of leading it back to turning into the skin cell, you make you encourage it to make certain gene expression decisions to turn it into something like a nerve cell, right? And then the quadriplegic patient, right, his or her spine can then regenerate. Right? And so that's the promise of it. So these transcription factors, they work together to decondense the chromatin, to expose DNA in promoter regions of genes that have been shut down a long time ago. They remove modifications to the DNA, right? And thus they restore the cell's ability to be pluripotent. And importantly, right, there is no concern of ethical or moral sourcing of these cells, because the beauty of this is you take a skin cell from the quadriplegic person herself, her own skin cell that contains her genome, right? And this is what you do to make a pluripotent cell that you then put back in her body so that it can hopefully heal her spinal injury. Okay, so early research into understanding the importance of the chromatin being condensed or relaxed, a lot of it was done in Drosophila, right, in flies. So flies have this interesting property where some of their cells, sometimes their ploidy, or the number of copies of their chromosomes, can be kind of really weirdly out of whack, right? They can have many, many copies of the same chromosome. And because it's many, many copies of the same piece of DNA, right? If you just had one copy of the chromosome, maybe this is kind of hard to see, right? But if you have many, many copies of it and they all kind of line up, you can actually see the features a lot easier. So that's what we see here. We have this polytene chromosome. Remember, this only happens in certain organisms like flies. We do not, this does not happen in our cells, but it's an important model to look at, because when these DNA copies of the chromosome are all lined up, it makes for a much thicker kind of molecule that you can see a lot easier under the microscope, right? That's what you see here. Scientists saw that when you have Drosophila cells that are busy doing gene expression, right, certain cells of the fly would be using certain genes but not other genes, they would see that along these chromosomes there would be these puffy regions, right? These puffy regions represent regions of the chromosome that are more relaxed. So instead of being really condensed, right, they kind of puff out. And if you zoom in to one of these puffs, this is just one strand of DNA from inside this big puff, they could actually see these bright spots. And what are these bright spots? These are RNA polymerase complexes that have bound to things like promoters of genes, right, that are now being transcribed. And that's why they saw that the RNA polymerase activity happens in these puffed regions, leading to the conclusion that, yeah, the control over condensation of chromatin versus relaxing of chromatin plays a crucial role in whether genes in the relaxed regions are now accessible to RNA polymerase. So what about in our cells? Right? I think I've already mentioned to you that in our cells, a typical, let's say, skin cell, fully differentiated cell that's no longer dividing, but it's just doing its normal cell type functions. Our chromosomes are not those thick rods that you see, in a karyotype, right? Normally, our chromosomes are very loose and they're kind of all intertwined, right? So this electron micrograph image is of an active nucleus. And the regions that you see here, this is all the chromosomes that are all jumbled together. I say it's like a bowl of Simon, right? Because that's how you can think of it, a bowl of Simon. Different regions of the genome take turns being condensed or more relaxed. It's a very dynamic kind of landscape of things changing, right? And hopefully what you realize is, yeah, that all has to do with where certain genes are in the genome that a cell needs at that given moment, right? Let's say in response to things like not enough oxygen, or in response to plenty of nutrients, right? Different genes located on different chromosomes need to be expressed for the cell to use them, right? So you would see a dynamically shifting landscape of dark spots, which are the more tightly packed heterochromatin, and the lighter spots, right, which is the more loosely sort of distributed euchromatin. And what that means is that if you could, for example, right, tell the difference between heterochromatin and euchromatin, right, in a particular cell, right, that's doing something, you might be able to identify what genes are the ones in the genome that actually are relevant to that cell doing something at that given time. Right? So how do you do that? Well, just like this Simon thing here, you can actually try to break up or digest the DNA, right? And just like you trying to bite the Simon that's dry right out of the pack, and you have a hard time doing that, right? You would have a much easier time biting the boiled cooked Simon. Right? And so that is a technique that researchers have used, right? Not actually eating them with their mouths, but they use certain proteins, certain enzymes that digest or break up DNA. Okay? So there's an enzyme called DNase, which by its name, hopefully you realize, it breaks up DNA. It chops the DNA strand. Right? So here's the DNase protein sitting on top of exposed DNA. And then this DNA protein is the right shape to sit in here. And it's the right shape to go ahead and it'll cut the double-stranded DNA there. Right? And so the more DNase activity you have, right, on a strand of DNA, the more chopped up the strand of DNA will become. It'll become just like little fragments. So then you take two different cell types. Since we were talking about Simon, I like those green packs of chicken Simon, those Sapporo Nichiban ones, whatever, right? My kids eat tons of those. Let's say you take two types of cells from a chicken, right? A cell that's going to form a red blood cell and a cell that's going to form an egg, right? And it turns out that certain genes that are relevant for this cell to differentiate into a red blood cell, right, are maybe just next door on the same chromosome as genes that are relevant for a cell to become an egg. Right, so this is just like the receipt, right? The salty snack coupon that I care about is a few genes away from the Maybelline Revlon Cosmetics coupon that my wife cares about, right? So I might want to use this one. Maybe my wife uses that one, even though they're on the same chromosome, right? So you can imagine in the red blood cell, that particular region where that gene is would be more unraveled, right? So that you can have gene transcription, gene expression happening from it. Right? So if you were to go ahead and treat the DNA from a developing red blood cell with the DNase 1, what would you find? Right? The relaxed parts of the DNA are easier for the enzyme to bite into and chop up. Right? The parts that are more condensed, right, the enzyme has a harder time. Okay? What about if we did the same thing using the DNA from an egg cell? Well, it might be an opposite situation where you have the gene that's more relevant to being an egg, right, being the thing that's exposed, and therefore that's the thing that gets chewed up by the enzyme. But the gene that's more relevant to being a red blood cell was protected from it. Right? So then you run these fragments of DNA on a gel, and you can really see. You can see the difference between the globin gene that's used in red blood cells versus the albumin gene that's used in the egg cell, even though right? These genes are maybe only a few genes apart on the same chicken DNA that both of these cells has. Okay, so we're out of time today, unfortunately. So I'll stop here today and we will continue with the slides that I didn't get to today, all right, in next lecture.